Hi, I am Gigi Kinney, and I will be presenting on the effect of Michigan wheat consumption on the gut microbiota. So an individual's gut contains trillions of microbes that form the gut microbiota. These microbes can protect the host from pathogens and regulate the immune function. But however, gut microbiota and dysbiosis can cause a disturbance in these processes, and this has been seen to be associated with many chronic diseases. So much research has been done on this to see how different factors can affect the gut microbiota, and it has been observed that dietary intake is a key player in managing the gut microbiome. More specifically, the Western diet, which is characterized by processed foods, has been seen to result in a gut and dysbiosis. In our study, we looked at how consumption of whole wheat affects the gut microbiome. Since grains make up a staple in the Western diet, it is important to research this area. Switching from a diet in refined grains to one in whole grains has been linked to improvements in the gut microbiome, which may be due to the fiber and polyphenol content. Since different types of wheats have different polyphenol contents, in this study, we viewed the effects of red versus white wheat on the gut microbiome, which should theoretically have different polyphenol contents. So we were looking to see how these different contents would affect the gut microbiome in our participants. This study took place across a 4-week time period, and each week, participants were to consume a full bag of crackers per day, which consisted of about 100 grams of whole grains. In weeks 1 and 3, the crackers were generic white wheat crackers, and in week 4, which was a test week, the crackers were made up of white whole wheat, and then in week 4, which was another test week, the crackers were made up of red whole wheat. At the end of each week, participants submitted a fecal sample and a fiber questionnaire, and the genomic DNA was extracted from each fecal sample and submitted for sequencing. So the sequencing results were analyzed for individual abundance and diversity, as well as specific changes between treatment weeks. So results shown in figure 2 show the enterotypes among each participant, and we can see that enterotypes stayed stable among the participants throughout the study, meaning that there was no major shift in the species present from week to week. Figures 3 and 4 take a look at the shifts in abundance of individual bacteria between treatments. So figure 3 shows the difference in species between weeks 1 of generic wheat crackers and week 2 consisting of white whole wheat crackers. So week 2 was the first week that we introduced whole wheat to the participants. However, there was no clear trend in the shift in abundance between weeks. And then we looked at the difference between weeks 2 and 4, which were the 2 test weeks. So in week 2, participants consumed white whole wheat crackers, and then in week 4, they consumed red whole wheat crackers. But again, there didn't seem to be a clear trend in shifts of the bacteria. The only species that differed significantly between treatments was lactobacillus. Lactobacillus, as seen, you can see it in figures 5 and 6, had a higher abundance in the generic whole wheat weeks, suggesting that whole wheat intake may reduce lactobacillus abundance. So overall, whole wheat treatment did not appear to have a significant effect on the gut microbiome, aside from the reduced abundance of lactobacillus from whole wheat intake. Additionally, the 2 treatments, white wheat and red wheat, did not appear to differ in their effects on the gut microbiome. There are many areas of research that have yet to be done with the data from the study. The fecal samples may be analyzed for biomarkers of wheat intake so that we can account for non-compliance within the study. Additional analysis may also be run to account for the BMI and socioeconomic status of the participants. So I want to thank Dr. Sugino, Dr. Garrow, Dr. Ng, Dr. Comstock, and the entire Comstock lab for all of their help with the study and with the analysis of the results. Thank you very much.